It's a pleasure to be here. And our lab works on sphingolipids. I'm going to talk in particularly about one pathway where we have a clinical trial concept. And it says, what are sphingolipids, and should I even care about these? And the answer to this slide is really it's all of the above. We are very interested because we feel that they are key signaling molecules and very important for cancer cell proliferation. We know that sphingolipids are part of the lipid bilayer of cells, and over the last 2 decades, ceramide has been shown to be a bioactive sphingolipid, which is involved in induction of cell differentiation, growth arrest, senescence, and it's known to stimulate neuroblastoma differentiation. And this is sort of what the ceramide molecule looks like. It's one of the key things is there's thought to be like a rheostat, like a balance between the amount of ceramide and sphingosine 1-phosphate, which I'll just call S1P within the cell. So if you have more S1P, you're going to have more inflammation, you're going to promote vasculogenesis, you're going to promote drug resistance, and it's an apoptotic molecule. Now, if you're able to shift that balance within the tumor cell and have more ceramide, you get the opposite. As you increase ceramide levels, you're going to promote more apoptosis. You may also, instead of apoptosis, cell cycle arrest, which will be important for stopping tumor growth and autophagy. So it's really, we want to shift this balance from S1P to more ceramide. The sphingolipid pathway is quite complex, and I won't get into it, but this is just a slide to show how complex it is. And what I'm going to be talking about is actually targeting this sphingosine 1-phosphate by using an inhibitor to sphingosine kinase 2. We know that, and the endpoint is really to downregulate S1P and upregulate ceramide. Ceramide has many effects, and it acts through ceramide-activated phosphatases, which in turn will cause dephosphorylation of PKC alpha, retinoblastoma protein, and in turn lead to cell cycle arrest and apoptosis. It's also known that ceramide is very important for to prevent the downregulation or breakdown of c-MYC and n-MYC as well. In our lab, we really focus on a number of enzymes, including the desaturase, sphingosine kinase, and other ceramides as well. Now, again, why should we target S1P? We really feel that by targeting this, we're going to block angiogenesis, block inflammation. There are roles in S1P being important for the immune system. And we know that in human cells, S1P is generated by 2 sphingosine kinases, sphingosine kinase 1 and sphingosine kinase 2, and it's really an attractive target for cancer therapy. Chuck Smith is in the Department of Pharmacy at our institution, and he gave a talk a couple of years ago where he found a compound, which I'll just call ABC. It's ABC294640. I'm just going to call it ABC. And he found this for a library screen, and then tested it in breast cancer, hepatocellular carcinoma, renal tumors, and he showed some great effects in cell lines, but also in mouse xenografts. It hadn't been tested in any pediatric tumors, and I talked to Chuck, can you give us some of the compound, and we'll test it. The nice thing, it's oral, it's very bioavailable, and it's currently in a phase 1 trial in adults at our cancer center, which I'm going to talk about. So we've done a lot of experiments, and this is just sort of a summary of some of what we've done, and we've looked at ABC alone and in combination with chemotherapy in neuroblastoma, rhabdomyosarcoma, and Ewing sarcoma. And this was data I presented last year at the meeting. This is in neuroblastoma cell line. This is an embryonal and alveolar Ewing sarcoma showing really nice synergy with doxorubicin. And we also did drug combination studies and looked at the combination index of Chuintalali, where drugs are at a fixed concentration, and then you treat, and if your combination index is less than 0.8, you have synergy. So around 1, it's additive. So we saw some really nice responses in all tumors, but especially in the sarcomas as well. But we thought about in patients, our patients have really been very treated with anthracycline, so we really don't want to use anthracycline as a combination. We really want to use the agents that we're using for salvage therapy. So we looked in KCNR cells, CHLA-20, CHLA-90, which are neuroblastoma cell lines, and in rhabdomyosarcoma and Ewing sarcoma, and this is the data here with topotecan, where in this cell line we get some nice additive effects, and you can see that's the blue is ABC alone, and then combination topotecan, we get a nice, we get more effects, and that's more pronounced in the rhabdomyosarcoma cell lines. That's the data with topotecan. That's our data with irinotecan as well. And again, we see some nice additive effects and in many times synergistic effects in combination in all the cell lines we've tested, which we're very excited about, and we'll be moving to xenograft models in the next couple months. We looked at it's supposed to affect ceramide levels, and this is just mass spec showing we get an increase in ceramide. We also saw an increase in dihydroceramides, which was a compound that lipid we're interested in, and we saw a nice decrease in S1P. We decided to look at, was it not only affecting the sphingosine kinase 2, but affecting the desaturase as well? And this is like an in vitro assay with red liver microsomes, and then the assay in KCNR cells, and we see nice inhibition of activity of the desaturase. It doesn't increase over time, showing it's an irreversible inhibitor, and it doesn't affect the level of the desaturase protein, so it acts directly on the enzyme. We've looked a little bit mechanistically as to what ABC is doing. In renal tumors and breast cancer, it's supposed to promote autophagy. We saw the same because we saw cleavage of LC3-1 to LC3, and we saw downregulation of AKT with dephosphorylation of phospho-AKT. These are some data from Chuck's lab in mice, and he showed, so it's hard to see that ABC is selective for SK2 and has minimal effects on other kinases, so that's very important. Then he also showed it's very bioavailable. These were mice which were given a dose of ABC, and then the levels were measured 30 minutes after administration orally, and we saw really nice, very bioavailable. And the half-life for the oral administration was 4.5 hours, meaning that could be given twice daily orally. Very importantly, the toxicity in mice was very impressive, where the only thing shown, panel A is 7 days, panel B is 28 days of treatment. So there was a transient decrease in hematocrit, not much of a change in your white count. Interesting, like a slight increase in your ANC. And at 28 days, there was the drop in hematocrit had resolved, and the neutrophil count had sort of stabilized. He didn't see any toxicity in terms of liver toxicity by CMP, any renal toxicity, and when they looked at autopsies or necropsies of mice, they didn't see any damage to any of the organs as well. So we were, very excited about this compound. And there's an ongoing phase 1 trial at our institution for adults with solid tumors. And it's a 2-part trial to 3x3 design. And the first part of this study is to determine the MTD in the adult patients. And then once the MTD is established, they're going to use that maximum tolerated dose for safety in patients with pancreatic cancer. And this is their sort of dose level. They're currently, as of last Thursday, at dose level 4, and they've just had their first DLT, so they're going to actually expand that cohort to 6 patients and then continue on. Our proposal is once the cancer center is very interested, and our proposal is once the adult MTD is established, is to start dosing the pediatric trial at 50% of the adult MTD. And what our proposal is, we don't want to do single agent. We want to have something that could potentially not only give us information on MTD, but really help our kids, really have some responses. So irinotecan temozolomide is one of our standard salvage therapies that we use for kids. And COG is using this combination sort of as a cassette to test new drugs. So we would use the same cassette that COG is using of irinotecan at a dose of 50 milligrams per meter squared, IV daily on days 1 through 5, and then temozolomide PO on days 1 through 5, and this will be repeated every 21 days with ABC given continuously. There are some nice data, some papers published that IV and oral irinotecan is very equivalent, but that's something that we could later discuss, and I think we want to sort of keep the same backbone that COG is doing, and we would open this for all solid tumors, would be our proposal for neuroblastoma, sarcomas, renal tumors, and CNS tumors as well. Chuck has told me that in some of my studies he sees some nice entry into CNS, so that could be a target. We haven't looked at any brain tumors per se. We would allow multiple relapse patients, not only first relapse, and our primary objective would be to determine the MTD, and then look at the pharmacokinetics, look at as biomarkers the plasma levels of the sphingolipids, and then also look at response. We in our lab, we feel that the sphingolipid target therapies have great potential for pediatric cancer. We hope to combine these with current chemotherapies we have available to overcome drug resistance. Many of them have additive effects, synergistic effects of radiation therapy, and many are known to inhibit tumor migration and invasion. And we hope that these therapies could potentially have less side effects than our conventional therapies. Really want to thank Giselle for bringing us here and forming this consortium. It's really such a wonderful group of people, and I really want to thank the parents and their families, because you are the inspiration for all the work that we do. And thank you again. I want to thank members of my lab who've done this work, our clinical team, who Amy Lee's going to talk after me. She's our new brain tumor specialist. Chuck Smith, who's developed this compound, Betsy Hill, and then the lipidomics core, and Whitney Ringler, Rich Ringler, and Adam White are here from Chase After a Cure. I said without their help, this lab wouldn't exist, and thank you so much.